So before I go on to the vaccine, I just want to point out some other things with some studies that I've been looking at, which are actually really vitally important to this whole thing. Many of you probably already know, but some of you probably won't. So the first study was from the CDC, and basically it brought up two problems. Well, I say problems, it just brought up two things that were very interesting. The first one is how they sequence the virus. Now, if there's a real virus out there, you'd be able to find it, you'd be able to sequence it end to end in a laboratory and say that that's what's causing the illness. But in this case, this virus is supposed to have 30,000 base pairs, and all they had from living tissue was 37 out of 30,000. The rest of it came from a digital viral sequence bank in China. So basically we're talking a computer 99% more of it computer generated virus. Right, that's vital to understand, because it's only done with a small bit of tissue. So it wasn't sequenced end to end with living tissue for a start. And this is what they're actually using. But the second thing, and this is the important bit, is that they tested it. They tested this SARS-CoV-2, the computer-generated virus, on 3 types of human tissue. Human adenocarcinoma, which is basically malignant tumour, human liver cells, and human embryonic kidney cells. And to do this, they inoculated a very high dose of the SARS-CoV-2 into the tissue, and then they waited 24 hours. And after 24 hours, they found there was no cytopathic effect. And all that means is structural changes caused by viral invasion to your cells. None of it. So basically it cannot replicate, meaning it's non-infectious to humans. The other thing they did, and this is the kicker, they told us that this virus started because somebody in China ate a bat. So they tried to infect bat cell lines with this SARS-CoV-2 that came from the computer-generated virus for replication capacity, and it couldn't infect the bat either. So we're talking about something here that's absolutely zero proof, scientifically or otherwise, that a virus even exists. There was another study that backed up the first part of the sequence coming from the lab in China, and that was done. There's basically 22 scientists who were asking for this study to be redacted immediately, down to 10 fatal errors and 4 minor ones. One of them is Dr. Kevin Corbett. And this study where they said they detected the virus was called Detection of 2019 Novel Coronavirus by Real-Time PCR. We've already spoken about the PCR, but this is the study that they're all using as a control. So this study was done within 24 hours, completely skipping peer review. That's unheard of, and this is why doctors are starting to speak out about this. Usually takes months to peer review. So, and it went through all, basically within 24 hours, and the PCR was accepted as the gold standard, right? And this is what everyone else is using. This is actually a quote from the study. We aimed to develop and deploy robust diagnostic methodology for use in public health laboratory settings without having virus material available. Without having virus material available. This is another quote from a study in Wuhan, no less. Virus cultures were negative for all asymptomatic positive cases, indicating no viable virus in positive cases detected in this study. They're actually telling us here, so if there's no virus that's been isolated or purified or even detected at this point, what on earth is in the vaccine? Because if you know how a vaccine is supposed to work, it's basically you put a small amount of the virus or a weakened amount of the virus into the vaccine, your body produces an antibody response so it recognises it next time. That's how it's supposed to work, right? Okay, so where's the virus? It's not in the vaccine.